So thank you once again for joining our third Thursday Food for Thought program. My name is AJ Von Arx, and I'm a volunteer board member and sometimes educator for Old Pueblo Archaeology Center in Tucson, Arizona, which is a nonprofit organization that is bringing you tonight's presentation. For tonight's presentation, we're delighted to welcome our guest presenter, archaeologist/geneticist, Dr. Jakob Sedig, PhD. Dr. Sedig is a senior archaeologist with the Chronicle Heritage Consulting Company. He lives in Cambridge, Massachusetts, but works remotely for Chronicle Heritage's offices in Phoenix, Albuquerque, and Ignacio, Colorado. Jakob received his doctorate from the University of Colorado, where he focused on the Mimbres traditional phase from 900 through 1000 Common Era. After graduating, he was awarded a postdoctoral research position at Harvard University's Reich Ancient DNA Laboratory from 2016 to 2018. And then he stayed on, and he worked as an ethics and outreach officer and research fellow. And that laboratory is located at Harvard Medical School in Boston, Massachusetts, and is officially called the Reich Laboratory of Medical and Population Genetics. During his time at the Reich Lab, he worked to fully integrate aDNA, or ancient DNA, with archaeological studies. He has been involved in using aDNA data to examine long-standing archaeological questions and to explore novel applications, including using aDNA to help refine radiocarbon dating. Jakob's work also has focused on the ethical issues of paleogenomic research, particularly those involving sampling methods and engagement with indigenous communities. In his free time, he says he enjoys playing with his daughter, singing karaoke, and taking his cat for walks, which, we all want to see video of that. This evening, Jakob will discuss aDNA data generated by a genetic study of over 300 individuals who lived in central, western, and northern Mexico hundreds and sometimes thousands of years ago. He will discuss what these Mexican population data may tell us about indigenous peoples across the Americas, and how this ancient DNA project has brought together archaeologists, geneticists, researchers, analysts, and students from different backgrounds and countries. We are pleased to welcome Dr. Jakob Sedig.
Hi, everyone. Thanks for the great introduction, AJ, and thanks for inviting me, Al. It's really my pleasure to be here and to talk to folks. So let me get started by sharing my screen and starting the presentation, which hopefully you can all see now. Okay, so some of you may remember me from 10 or 12 years ago if you were part of Old Pueblo Archaeology's visit to Woodrow Ruin. So as AJ mentioned, I worked at Woodrow during my dissertation research when I was a graduate student at University of Colorado. And at the time, I had no idea that I would be pursuing ancient DNA research. My career would take a pretty big detour into that field. So if you saw me while I was at Woodrow, you saw me dirty, digging holes in the ground, which I don't get to do nearly as much as I'd like to anymore. And my research then was focused on the Mimbres transitional phase. So this period of time between the late Pit House period and the Classic period, and especially sort of the numerous social and cultural, religious transformations that occurred in the Mimbres region during that time.
Excuse me, Jacob, can you go to
Yeah.
15?
Oh, are you seeing my notes, or are you
Yes, we are.
Oh, okay. All right. Then let me try again.
There you go. Well, we can see you now.
Okay, yes. I will share again.
It's a good view of you, though.
Okay, so now,
Out of full screen from beginning.
yeah. And you're probably seeing my notes again, right? I have an extra monitor. Let me unplug that and see if that does the trick. You still seeing my notes?
Yeah.
Okay.
We saw a flash of the one.
Okay, let me try it this
Oh,
way.
now we have it. Okay, now go full
Yeah,
screen there.
there we go.
Excellent, thanks.
Okay. Okay, great. Sorry about that. You would think after 4 years on Zoom, I would have these technical difficulties ironed out, but
No.
not so. Um, okay, so yes, I spent some time in Mimbres region, thought that's what I'd be doing for my career, and I still am a Mimbres archaeologist, but I took a major detour into ancient DNA, and I'm glad I did. This is a really new and exciting field that's helping us understand all sorts of things about the past that previously weren't possible. So how did I go from digging holes in the ground in southwest New Mexico to giving talks in Honduras and Guatemala about ancient DNA and publishing a number of papers about ethics in ancient DNA, and then also research papers? Long story short, when I finished my PhD, I moved out to the East Coast with my wife and cat at the time, Woodrow, who Woodrow still lives here with us on the East Coast. And through networking, got into touch with David Reich, the PI at the Reich Lab at Harvard University. So when I first met David, he was looking to bring on an archaeologist part-time to help the lab build connections between archaeologists and establish relationships with museums and other researchers and communities. And then in 2018, starting in 2018, David got a big multi-million-dollar grant, and at that time brought me on as the a postdoctoral researcher and an outreach and ethics officer in the lab. And that position, that position ended last December. So I spent close to a decade working with the lab, and I still am a research affiliate with the lab. And during that time, the thing that I've been saying is that I basically got a second PhD. I don't have a diploma, a piece of paper to prove it, but I had to learn genetics. I had to learn ancient DNA. That was not something that I focused on when I was, completing my PhD at the University of Colorado and analyzing Mimbres ceramics and field data. But in order to help the lab build those connections and establish those relations, I had to learn as much as I could about genetics so that I could explain it to non-experts. So, over the course of those years, I've picked up the ability to do some analysis on my own. I'm not like one of the geneticists in the lab. I can't do the full suite of really complex analyses that some people can do, but I know enough to study basic population genetic questions, and maybe more importantly, explain what we do to archaeologists and local communities and museum curators, the people who aren't experts in ancient DNA. And I wanted to mention before I'm gonna spend the next little part of my talk getting into just some key things that I think people in the audience should be aware of ancient DNA, the techniques, how they work, and terminology, because this still is a new field, and this may be the first talk some of you are hearing about ancient DNA. Before I do that, I did want to mention too, I'm going to be talking about human remains and extracting DNA from those remains. I don't use images, skeletal images of human remains in the talk, although I do use some illustrations. But I just wanted to note that because I know this can be a sensitive topic for some people, and I just want to make sure everyone was aware of that. So ancient DNA, let's start at the basic term itself. What is ancient DNA? When we say ancient DNA, what we're referring to is DNA of organisms that died hundreds, thousands, or even millions of years ago. And there's not really an age cutoff for something to be considered ancient. I've worked on projects where we study individuals from the 1800s. So that would be historic if we're thinking of things in terms of chronology or archaeological terms, but we still refer to the DNA of those individuals as ancient DNA. Sometimes you'll see the word people use historic DNA, but generally we refer to all DNA from any individual that died at any period of time as ancient DNA. And so really when we say ancient DNA, what we mean, what we're talking about are the the techniques, the set of tools that we use to study and isolate DNA from an organism that's no longer living. Doing ancient DNA research is really complex. This is one of the things I learned over the years. It's not, I won't say doing radiocarbon dating is easy, but as an archaeologist, it was something I could pick up and understand sort of intuitively pretty quickly. That's not the case for ancient DNA. There's so many steps involved, and there's so much biology involved that it was totally foreign to me. Ancient DNA, when we study it, when we obtain it from an ancient organism that is either in a burial context or environmental DNA, there's a few things that we do to authenticate it as ancient DNA. So if you think of DNA, probably what you think of is the long double helix chain, 23 different chromosomes. But that's not what DNA looks like as it starts to degrade. As it starts to degrade, it gets divided up into smaller and smaller chunks. And one of the big things that we have to do is reassemble those chunks, or not reassemble them, but organize them so we know where in the genome we're looking at. There's a few things that we can do as first steps help us determine that what we're looking at is actually authentic ancient DNA and not contaminating DNA from a modern individual. That's what this slide is meant to get at. So as DNA degrades, there starts to be mutations. Mutations occur on those strands. It's almost like fraying if you think of a string start to fray at the ends as it ages. That's a pretty good analogy for what's happening. The exposed edges of the strand, mutations start to occur there. We can quantify. We can look at those mutations, and seeing them helps us identify DNA as authentic and not authentically ancient and not modern contaminating DNA. So how do we obtain ancient DNA? As I mentioned, we're now at the point where it doesn't just have to come from bone. We're able to obtain ancient DNA from the environment, from the soil. There's a really interesting paper a few years ago now, maybe even 5 years ago, where researchers extracted DNA, human DNA, from a tobacco pipe, a historic tobacco pipe. So we can extract ancient DNA from many, many different materials. But with that being said, when we're talking about human DNA, the majority of it comes from bone. And so what we have to do is take the bone. Typically, this involves sampling it. We've developed a number of methods to really minimize the amount of damage that we have to do to a bone. We're almost to the point we need less than 50 milligrams. So the tip of a pencil is about as much material that we need. But we turn portion of that bone into powder, extract DNA from that powder, and then do a number of steps, which I'm going to talk about next, to help us isolate and amplify the authentic ancient DNA so that we can study it. And then I also wanted to note that the Reich Lab, where I was a postdoc and I'm still a research affiliate, is one of the leading ancient DNA laboratories in the world. In fact, we've generated over 50% of all ancient DNA samples that have ever been published. Most of those come from Western Europe, but increasingly other places of the world are being studied too. This next slide, I wanted to walk through the ancient DNA pipeline a little bit, so you have a sense of the steps that we have to go through in order to make ancient DNA data that can be studied. So as I mentioned, we start with bone typically, although it doesn't have to be bone. The next step is to take that sample into one of our clean rooms, and this really is like something out of science fiction. When I first saw it, you go into the Harvard Medical School. You need a security card to get in, and once you're in, you can't actually enter the clean room because we want to minimize contamination from modern DNA. But you can look into it, and when you look in, there's an eerie, bluish UV light that's shining in the room. And the reason we do that is because whenever there's not a technician in the lab, we bathe that wet lab in UV light so that it kills modern contaminating ancient DNA. There's a number of other features that the wet lab has to help minimize contamination too. The air only flows one direction, so it's constantly circulating fresh air into the room. When a technician enters the wet lab, as you saw on the last slide, they have to wear full Tyvek suits and face masks and gloves. During COVID, one of the safest places to work was actually the wet lab because the technicians, well before most people were thinking about PPE and that type of equipment, the wet lab technicians had to be fully garbed in those outfits to keep their DNA to themselves and not contaminate the ancient samples. So after the sample is in the wet lab, it's turned into a powder, and then we do a number of steps to turn that powder into an extract, and then from an extract to the library. I won't get into the details of this, but basically what we're doing is dissolving all the bone mineral so that all we're left with is a soup of DNA. Now, this DNA soup has not just human DNA initially. It'll have the DNA of all the other organisms that came into contact with that bone or whatever sample that we're processing originally. So this could include things like bacteria, microbes, contaminating DNA. And again, there's a number of steps that we take to eliminate those and really amplify and focus on the authentic human ancient DNA. Once we have that, we send it to a sequencer that helps us align the different DNA fragments. So we know what part of the genome we're looking at. And then once we have that data, we can start to do our analyses, the various analyses. Now, I'll say my job in the lab, I did some of the front end work, obtaining samples and bringing them to the lab. And then, as I mentioned, I could do some of this back end work, the statistical work, but all of this work is done by other specialists. So there's wet lab technicians that process the bone samples and then do a number of steps to extract DNA. Bioinformatics people process the data that we have and prepare it for the analysts in the lab, people like me who can look at the data to help us understand ancient individuals and the populations they were a part of. So just a few more terms that I wanted to mention because I'll throw them around and I wanted to make sure everyone's familiar enough with them so that when I use the terms, they don't sound totally foreign. So endogenous DNA, that's the authentic ancient DNA from a particular individual that we're looking at. So I just mentioned that DNA soup that we create. At first, it has not just the ancient DNA from the individual we're interested in, but anything else, any other organism that individual or those human remains may have come into contact with. And you can see it's actually a small percentage of that DNA soup. The endogenous DNA that we're interested in is only a small percentage. There's non-target endogenous DNA. So that's DNA that is part of what's sometimes called junk DNA. So our genomes are about 4 billion base pairs long, and the majority of that is shared between every person that's alive and isn't really that informative. We don't target that in our work because it wouldn't really tell us that much about a particular individual. But you also have things like environmental DNA, modern contaminants, and then little bits and pieces of DNA that are maybe only 3 or 4 bases long that we can't identify because there's not enough there to help us determine what organism it may have come from. So another term that I'm going to use is SNP, or single nucleotide polymorphism. So these are the individual A, T, C's, and G's, adenine, thymine, cytosine, and guanine, if you remember your high school biology class, that make up our DNA chains, those over 4 billion base pairs that I just mentioned. So as I said, most DNA that we have is not that informative. It doesn't tell us that much. It's really only a small portion of our DNA that accounts for all of the phenotypic human variability that we see. And those are the SNPs that we're interested in. For most of my time in the lab, there's about 1.24 million SNPs that we would study. But in the last couple of years, we've increased that to now we're working with 2 million or even 3.2 million different SNPs that we can analyze. And again, these are the variable positions within the genome that are most informative about an individual and what differentiates, an individual or a population from other individuals or populations. And then one last thing, mitochondrial DNA versus nuclear DNA or whole genome DNA. So mitochondrial DNA was the first DNA that was obtained and studied, what we might call ancient DNA. I think the first ancient DNA study was from a horse, and it was, I want to say 1984 is when that was first published. So mitochondrial DNA, it's DNA that lives in an entirely different part of the cell, in the mitochondria, and it is a much shorter DNA chain than what's found in the nucleus of the cell. So mitochondrial DNA is 12,000 or 14,000 base pairs long versus 4 billion for nuclear DNA. And it's only passed down from a mother to her offspring. So I have my mitochondrial DNA is the mitochondrial DNA of my mother. I didn't pass that mitochondrial DNA down to my daughter. My daughter got her mitochondrial DNA from her mom. The reason why mitochondrial DNA has been studied, we were able to study mitochondrial DNA sooner than nuclear DNA, is because it is so much shorter, so there's less to process, but also there's much more of it. It's easier to obtain. So I'm going to talk a little bit about previous mitochondrial DNA studies later in the talk, but just know for now that this was sort of the one of the early ways that we could study ancient DNA of individuals that lived at some point in the past. Okay. So hopefully now you all have a sufficient enough background with some of the terminology I'm going to be using and how we use the techniques we use to obtain ancient DNA, that I'll be able to not sound I won't be using too much jargon when I talk about the next segment of my presentation, some of the really interesting work that we've done. So I'm going to be focusing on the PIPANOM project, what we call the Pipnom project, which stands for Proyecto de Poblaciones Antiguas en el Norte y Occidente de México. And in case you don't speak Spanish or understand my very bad Spanish, that basically stands for project of studying ancient populations in the north and west of Mexico. So this is a really exciting project that's been years in the making, and we're getting closer to publishing. We had a number of setbacks with the project, notably COVID, but it's this large-scale project that involves archaeologists and geneticists from across Mexico and the United States, that the goal is to acquire samples from hundreds of ancient individuals so that we can really start to study what those ancient populations, what their structure was like, how different populations were similar or different to one another, and particularly interesting things about individuals within those populations, which is what I'm going to spend some time talking about next. For me, one of the most exciting parts of the project, along with the science, is the outreach and the engagement that we've done. So with our Mexican collaborators, we've held a number of events open to the public, and my favorite school kids who have had a chance to come in and learn about ancient DNA, and the really interesting things that we can learn about the past by studying it. And I think it's this type of outreach that we really, as scientists, need to do more of to help get the next generation interested in our work, but also share the results of our research with the broader public. The Pipanom project spans thousands of kilometers across, as far north as you can get into Mexico, into western and central Mexico, around, about as far south as Mexico City. And we cover a variety of cultures and time periods. So this slide just gives you a sort of a brief overview of the different archaeological cultures that we're working with. So we have everything from big Tarascan sites, Tzintzuntzan. I've been fortunate enough to travel there a couple times now as part of the Pipnom project. And if anyone ever gets a chance to go there, I highly recommend it. I've never been to an archaeological site like Tzintzuntzan before. And this is the most densely sampled site that we have in the project. We have over 100 individuals from Tzintzuntzan. But we also have, individuals, dozens of individuals from the Chalchihuites culture and Aztlan and Trincheras individuals and Sonora. And then, of course, individuals from Paquimé or Casas Grandes in northern Mexico. So I'm going to spend some time talking about the results that we have from Paquimé now, and this one individual in particular. Here's a breakdown of the results that we have. And maybe the first thing that you've noticed is that most of the individuals that we sampled, that we tried to acquire ancient DNA from, didn't work. This is a major disappointment for us. But even though we weren't able to get data from those individuals, it was disappointing, but it wasn't too unexpected. And there's a few different reasons for that. In general, ancient DNA doesn't preserve well in hot, arid environments, which, if you've ever been to Paquimé or Casas Grandes, you know that's certainly the case there. But then, notoriously, many of the individuals that were found or excavated from Paquimé were not buried. They were left on the surface of the site at the site's closure, the site's abandonment. So if an individual is buried in the ground, their skeleton is protected from some of these elements, but if they're on the surface, they're exposed. So DNA will degrade more rapidly. And then finally, after these individuals were excavated by the Joint Casas Grandes project, Charles de Peso, in the late 60s and early 70s, they were stored in basically a shed with a metal roof that was not air conditioned, and they were on the top shelf of that in that shed. So they were basically in an oven for 60 years, more than 60 years, and that really degraded the DNA also. So again, it's disappointing we weren't able to get working data from those individuals, but not that unexpected. With that being said, the fact that we were able to get working data from 54 different individuals is really that's something that we're proud of, and that's a decent enough sample size for us to really get into some interesting questions about Paquimé and the people who lived there and how they related to nearby and contemporaneous populations. This next part of the slide is showing what skeletal elements we sampled. You can see that somewhat surprisingly, petrous, the inner ear bone, which tends to work best, it tends to preserve ancient DNA better than any other skeletal element, had a lower success rate than the teeth. But I think that's because we had just a lower sample size. And if we boosted up this sample size, I would expect that the percentage of petrous samples that worked would be, similar to teeth or better than teeth. And then postcranial bone, we only did a few of those. They actually, that small sample size worked pretty well too. So again, even though the majority of the ancient individuals didn't produce working data, we were still able to have a good working data set from Paquimé. And our mean SNP count, there's that term again, single nucleotide polymorphism, was about 200,000, which is plenty enough to work with and do our standard population genetic analyses. So we took those data from those, 54 individuals plus 11 who had, marginal data, and combined that with our Pipnom data set, and then basically all other data that has been published from the Americas to have a working set of over 500 ancient individuals. And, we're really now starting to explore some interesting things, long-term archaeological questions about these cultures and the people within those cultures that were previously much debated, but didn't have definitive answers. Okay, so now I'm getting into, I think what Al was mentioning at the beginning, some of these slides looking really dense and technical. I don't expect you to be experts looking at this, and hopefully I'll do a good enough job explaining what's on here. I will say my 4.5-year-old daughter likes this slide because of the colors. If you ask her what her favorite color is, she'll tell you rainbow. So this is, it's very much a rainbow slide. To walk you through this is our basic analyses that we do in the lab whenever we generate data from an individual or a group of individuals to help us understand what their ancestry is like and how they're situated in sort of the broader regional context. So we have a map here of all the published ancient individuals from the Americas along with the Pipnom data set. What we have in these next two panels, B and C, are a PCA, a principal component analysis, which is one way that we can see how an individual clusters or is more closely related to other individuals than others. So B is just zoomed out and panel C is zoomed in. And what you see here is that this one individual who I keep hinting at from Paquimé, a child who was buried in the House of the Well, if we look at that individual's population level genetics on the PCA, there's nothing here that we wouldn't expect. So he on the PCA is clustering with other individuals from northern Mexico, exactly where we would think someone who is buried at Paquimé might plot on a PCA. This next panel below is what we call admixture analysis. It's another way that we look at a person's ancestry. What we do with admixture analysis is run a program and tell the program to split an individual's ancestry into different components. We tell the program how many components we want to analyze, and we typically do 2 through 15. And then we can there's a way that we can determine which number of ancestry components are most likely reflective of what an individual or a population's ancestry might look like. So what you can see here is this individual from Paquimé burial 23-8. The admixture looks, again, like you would expect for someone from ancient northern Mexico. There's a lot of this turquoise component with lesser amounts of the other components. These other sites that have a lot of green are like Tiopa, Sonora site, Chihuahua Middle Ages, some Trinchera sites. You see increasing orange as you move to California, and then increasing purple as you move south. Again, nothing unexpected. And then if we look, this is what we call F3 statistics. And with F3 statistics, all we're doing is taking an individual or a population, comparing it to a test population, and then an outgroup, a group that both of those populations or tests should be sort of equally related to. So typically that's a group like Mbuti or an African population. And then we just run a number of tests to see how closely related those two groups are or how much allele sharing they have. So in this one, what we're doing is we're taking this burial 23-8 and comparing it to a number of other populations that have been previously published or are part of the PIPANOM project. And again, what we see is that this individual from Paquimé has ancestry or allele sharing most similar to other individuals from northern Mexico. So far, so good. When we're looking at data from this individual, there's nothing suspicious. But then we did a new analysis called runs of homozygosity. So this is a relatively new technique to ancient DNA that we just integrated into our analysis pipeline a couple of years ago. This slide is maybe even more technical than the previous one. Again, look at the pretty colors. I'll sort of explain what's happening here. But basically, runs of homozygosity are parts of a genome that are repetitive. So if you've had high school biology, you know that you inherit one chromosome from your mom and one chromosome from your dad. If your mom and your dad happen to be closely related, say brother and sister, they're going to have a lot of shared or overlapping DNA. And so if we study, if those two individuals happen to produce a child and we look at that child's DNA, there's going to be lots of repetitive chunks in that child's DNA because his parents were closely related and had similar DNA. And so this is what these slides are sort of getting at here and how we look at this in our analyses. And so, for example, here you can see the two chromosomes that this individual has. There's a lot of overlap, zeros in the same place, ones in the same place. And these are just the different ways that we quantify that. So we ran the runs of homozygosity on this individual from Paquimé, as we did all individuals, and something jumped out immediately. This burial 23.8 had a very elevated level of runs of homozygosity. And what we see here on the left is just another way that we quantify runs of homozygosity. What we do is we take those chunks that are repetitive and sum them up, and we can those chunks we divide into small, medium, or long sections. They tell you different things. We're really interested in the long sections here. The longer sections, the longer the section, the more closely related an individual's parents were. And you can see here, this is what we would expect to see if an individual who had parents that were first cousins. This is what we would expect to see if a person had parents who were second cousins, third cousins, etc. And this individual from burial 23.8, I remade this figure with the values for burial 23.8 year, had parents who were clearly more closely related than first cousins. Not as closely related as siblings, but in between siblings and first cousins. So this is what we would call a second degree relationship. So siblings and parents and offspring are a first degree relationship. Relationships like grandparent-grandchild or half siblings would be second degree relationships. And that's what the ROH values we have are for this individual, burial 23.8. So that was really surprising to see a level that high. I think it was about 270 centimorgans. So we looked at all the other individuals that have been published and in the Pippanom data set. And what do you know? If you pool them together, most of the published individuals have long ROH runs that run somewhere between 0 and 125 centimorgans. And I'll say the majority of those are at the low end between 0 and 50. A significantly fewer number of individuals have, long ROH values that would be at the first cousin level. And then there's only one individual that's been published so far in the Americas that has a value this high, burial 23.8 from Paquimé. So once we saw that, we, immediately started digging into the metadata. And that was interesting too. So this individual, burial 23.8, comes from room 21 in the House of the Well. The House of the Well is one of the more well-known structures within Paquimé. It's full of the trade goods that you've probably heard about if you know anything about Paquimé shell and other items. But also it housed an underground well, a subterranean well, where we think a number of rituals and ceremonies occurred. So this room 21 in the House of the Well, it's adjacent to the well itself. It's not in the well, but it's adjacent to it. And burial 23.8 was found underneath the post, wrapped around a post beneath the floor in room 21. if you know, if you're a person who's interested in Paquimé, you probably have heard of this burial before this talk. This is a pretty well-known burial. It's the only burial in Paquimé that's like this. So this is a child aged somewhere between 6 and 10 years old, wrapped around the base of this post. On top of a sandstone disc, and underneath that sandstone disc, there was some turquoise offerings. That wasn't the only interesting thing about the burial. According to De Peso, there's damage at the back of the skull, which is suggestive of a blow to the back of the head, which led De Peso to think that this child was some sort of supplicatory offering, a sacrifice, basically, that was made during the Paquimé phase remodeling. Now, De Peso, his chronology at Paquimé, we found later, wasn't exactly accurate, but some of the work that we did with this individual, the radiocarbon dating, which I'll get to next, well, I'll just jump ahead, shows that this individual lived, right at the height of Paquimé's occupation during the Medio period. Going back a slide, once all of these pieces started coming together, we did a number of analyses. We did strontium isotope analysis. This helps us determine where this individual lived. So we got a value. We compared it to published strontium isotope values from Paquimé, and burial 23-8 is basically smack dab in the middle. So exactly what we would expect of a local, someone who lived and died at or near the site during their lifetime. So the picture that we have at this point is a child in a really unique burial context who had parents that were either, grandparent, grandchild, aunt, uncle, niece, nephew, or half siblings, born and died at the height of Paquimé's occupation, and lived locally his short life there. But we wanted to do some other research to help us really contextualize what was going on. There's really the first paper in the field of ancient DNA that looked at runs of homozygosity. Was a study at Newgrange, a Neolithic passage tomb in Ireland, really cool site. And they found an individual who had parents who were siblings, so even more closely related than Paquimé. But again, our individual from Paquimé, although his long ROH values are less than Newgrange, those values are still higher than any other published individual from the Americas. And interestingly, for Newgrange, the authors argued that this individual who had parents that were siblings was part of a dynastic lineage, that it was basically elites who were interbreeding with each other as a way to solidify their power. We did some more digging into the literature. Like I said, ROH is a relatively new technique. It hasn't been around that long, but there's been a couple studies. This is from the paper that really introduced the technique and the program that we use to study ROH. And this paper studied many thousands of individuals, ancient and modern, and looked at their runs of homozygosity. And out of this global data set, there was only 2 individuals that had ROH values higher than burial 23-8. And in fact, only 71 out of 1,941 individuals had values that were more than first cousins, greater than 50 centimorgans. So overall, this is suggesting that close inbreeding is actually pretty rare in the archaeological record. The author, Harold Ringbauer, who was also a postdoc in the Reich Lab for a while, an earlier study also looked at ROH in some Peruvian individuals and found some elevated levels, not nearly to the extent of the Paquimé individual. And they argued in that paper that this practice was part of a society that was what they classified as a high chiefdom. So again, elites were partaking in this interbreeding practice. And overall for the Americas, again, just to reiterate, we do see long ROH. Maybe every once in a while you'll find an individual who had parents that were cousins or second cousins, but not anything nearly as high as burial 23-8. We also looked at the ethnographic record to help us understand this and contextualize this. This probably isn't surprising, but we know that inbreeding is almost nearly universally a taboo. Rarely do you find mating pairs that are more closely related than second cousins, but there are some exceptions to this, some well-known exceptions. You know, ancient Egypt, Egyptian elites are, you know, probably some of the best-known examples of this. But the point is, it's really in these elite societies or hierarchical societies where we find these exceptions. And the reason for that is elites are allowed to break taboos. They like to make rules for the common populace, but then the elites can show their power by breaking their rules, you know, and saying, you're not allowed to do this, but I am. And so that was one of the takeaways that we found, you know, combing through the ethnographic literature, is that this practice does tend to occur more commonly in you know, hierarchical societies. It's a strategy that elites use. So I had to put this in here. This paper has gotten a pretty decent amount of press. I think I forgot to mention the paper was published in August and in the journal Antiquity. And when it was published, you know, some of the press picked it up, and they of course related it to Game of Thrones. For people who've watched Game of Thrones, there's some incest that happens in that show. But interestingly enough, it's elites that are participating in that inbreeding in Game of Thrones. So to sum things up for Paquimé, what we found is that burial 23-8 has a unique burial context. Had parents who were more closely related than anyone else examined at Paquimé, and in almost all of the published ancient DNA data up to this point. We feel that this is indicative of elites partaking in some sort of inbreeding practice to maybe solidify their power. And then this child was sacrificed as perhaps part of a ritual meant to consecrate this important building, this House of the Well, that we know a number of ceremonies and rituals was held there. So that's just one case study of one interesting individual from Paquimé that shows the power of ancient DNA and what we're starting to learn from it. I'm coming to the end of my talk. I did want to go over some other just basic results that we have from the Pipnom project at this point. I will say these results are preliminary. I'm fine with this video being recorded and shared and people, you know, discussing these results outside of this talk, but just please note they're preliminary and things might change as we continue to refine our analyses. So here's the Pipnom data set in large. You can see all of the individuals that we've studied now from, you know, southern, central, and northern Mexico, and previously published data from the Americas. I'll note here's Paquimé, basically right here. So again, what we would expect in burial 23-8 is somewhere in here. This is what we would expect to see. You know, this is sort of the typical structure of populations across Mexico. There's a gradient that flows with geography. I'm going to take a couple little detours here. When Al invited me to give this talk, he mentioned a previously published paper from 15 years ago or so now that looked at mitochondrial DNA. If you remember back to the beginning when I was talking about mitochondrial DNA, from the Mimbres region of New Mexico. And what this study found was that haplogroup B, mitochondrial haplogroup B, is the most common in the Southwest, and there's lower frequencies of A, C, and D. So a haplogroup is basically, it's a way for us to describe the particular mitochondrial DNA of an individual. So B is more common in the Southwest, A is more common in Mesoamerica, and there's one individual from Nan Ranch who had haplogroup B, but there are some mutations in the haplogroup that are common with present-day Tarahumara. So that was interesting. You know, maybe that's evidence of people from the Mimbres region interacting with groups further to the south. I wanted to note, though, this is our breakdown of the mitochondrial DNA haplogroups at the 50 plus individuals from Paquimé that we've studied. And you can see here, haplogroup B and some of its derived subgroups are by far the most common. Almost 75% of all the individuals from Paquimé are B. A couple of individuals, haplogroup A, that more Mesoamerican further south haplogroup, and then a couple of D and C individuals too. So the fact that we have an individual with these Mesoamerican derived mutations at Nan Ranch in the Mimbres region, to me, isn't that exciting or not that unexpected. It's exciting, it's interesting, but it's not that unexpected. So if I go back to that first slide with the Pipnom results, like I said, it's what we're seeing is a gradient. And so you would expect, some individuals to maybe have some of those Mesoamerican mutations, but that doesn't necessarily mean that that individual was from Mesoamerica or from southern Mexico. The data that we've looked at least in these northern Mexican sites, there's not any outlier individuals that have a clear, genetic signature like they were from somewhere further south in Mexico. I also wanted to note another really interesting paper that I was an author on. That came out last year, almost a year ago exactly. This was actually the first Pipnom data set. Some of the individuals from the Pipnom data set that were published. So they were published in conjunction with a number of ancient individuals from California. And I should note with the reason and we were able to study these ancient individuals from California was because there was extensive outreach and engagement with tribal communities there. And in fact, the first two authors on the paper are from tribal groups. In particular, the second author is from the community that we studied. And this is the way forward in ancient DNA, conducting these studies, especially in the United States. This is the work that has to be done before these types of studies can move forward. And it was really great to work with those communities and share the results with them. But the interesting thing about this paper, just to sum it up, because I'm running short on time, is what we found is that these individuals from California, what we have here, the oldest individuals really had ancestry that was specific to California. But then as you move forward through time, there's an increase in Mexican ancestry that particularly looks like, individuals from Cueva de los Muertos Chicos and Tayopa. And if you note the date here, this starts at 5,000 years ago. So this predates what we typically think of as the spread of maize agriculture into the US Southwest. One of the traditional models is that maize and the Uto-Aztecan language spread north from Mesoamerica into the Southwest and into California, at the earliest 4,000 years ago. But what we're actually seeing is that Mexican ancestry began increasing in some of these California individuals as early as 5,000 years ago, so predating the spread of maize agriculture. So that's a really interesting result. And again, it's something that is one, helping us understand better these ancient populations and how they related to one another, but also providing more insight into long-standing archaeological questions. I am running short on time, and I have many slides left. And I think for everyone's sake, instead of me trying to rush through everything, I will just skip ahead to my acknowledgements. Oh, okay, I can't. This is an interesting one. I'll stop here and talk briefly about this. I mentioned F3 statistics before. This is another way to visualize F3 statistics. So what we have here, what we're doing is testing these different populations. So say Paquimé against, previously published individuals from Chaco, previously published individuals from Cueva de los Muertos Chicos, and then individuals from the Pipnom data set from the north, the earliest individuals a little bit later in time. And what stands out here when we look at the entire data set is that these northern individuals and really are more closely related to each other than some of the other groups. Like if you go down to where we're comparing the different West groups to one another, you don't get, that dark blue like we see in a cluster here. So to me, that's potentially indicating that these groups in northern Mexico and Sonora were more isolated and secluded than some of the further south and further west populations that we've studied as part of the PIPANOM project. Here you can see the Paquimé F3 statistics highlighted here.
Excuse me, Jacob.
Yeah.
We scheduled this program for an hour and a half in case you
Okay.
go over an hour talking, so please feel free.
Okay, yeah, I'll talk a little bit more. I know some of this stuff can get pretty dense, so I'll talk a little bit more, but then I think I'd love to do some Q&A too, because
Yeah, I'd
rather
like to
than
make
me,
time for that.
yeah. So yeah, I'll just talk a little bit more. I don't have too many slides left. So this is yet another way that we can visualize F3 statistics. This is called an F3 tree. This is maybe one of my favorite ways to look at those F3 statistics because it helps us see how these populations, form a tree, how closely related they are to one another. So, what you have here for Paquimé, what you can see is that Paquimé is more closely related to, individuals from northern Mexico than, say, the Maya area, which is not, that's not surprising. That's what we would expect. I haven't talked about what some of these labels mean yet. You'll have to wait for the Pipnom paper when that comes out. This is basically shorthand. What we did is group the different geographic regions and time periods together. So when you see something like North 4, what that means to me is that these are the individuals from the northern part of the Pipnom project, and moving through time, 1 being earliest, 5 being latest. So these are North 4 individuals. So these are individuals that are basically from the Medio period in Paquimé would be North 4. Although in this particular F3 tree, we've separated Paquimé out. Okay, one other statistic I'll briefly mention is an F4 statistic. It's another way for us to understand and study how different groups are related to each other. So when I described F3 statistics before, I mentioned what we're doing is comparing 2 different populations to each other and then to an outgroup, just because they should be sort of equally related to that outgroup. With F4 statistics, we've added another group. So what we're doing is taking 2 populations and testing them as a clade, basically saying, are they more closely related to each other than they are a test population, this third group here. And depending on the result that we get, we can either say, yes, population 1 and population 2 form a clade, a branch off this tree, and they're distinct from this test population, or again, depending on the z-score that we get, the result, actually population 2 is more closely related to a test population than it is to population 1. There's a number of ways that we can visualize this. This is one way. This is from a paper that was published in early 2022 from the Maya region that looked at some really early individuals from Belize, cave sites in Belize. And what you have here along the y-axis is proportion of Chibchan ancestry. So Chibchan is South American ancestry. And what we see here is that the earliest Maya region individuals, so from 9,500 years ago till about 7,500 or 7,000 years ago, there's less of that South American Chibchan-like ancestry. But then as we move forward in time, that Chibchan-like ancestry increases, it shoots up. And the hypothesis in that paper is basically maize was re-domesticated in South America and then brought back from South America into the Maya region. And that increase that we see in South American ancestry, Chibchan-like ancestry, is evidenced here. That's why we're seeing that increased ancestry, is because it's coming with maize back from South America. So for our Pipnom data set, there's so many different tests that we can do. When I run F4 statistics, if we're looking at 20 different populations, let's say, so again, like we have West 1, West 2, West 3, West 4, West 5, North 1, North 2, North 3, North 1, 2, 3, 4, 5, and then, some other subdivisions that we might have. And then you have to run the test with each of the varieties of those. You can get thousands upon thousands of F4 statistics, which it's computationally is not that difficult to do. So I've done that for the Pipnom project. I've put, the Paquimé results here because it's relevant to what I've been talking about. And so what I did with this test is split the Paquimé individuals into the Medio period and the Viejo period, because one of the longstanding questions we have about Paquimé is whether the population there, the growth explosion that we see during the Medio period was the result of migrants moving into Paquimé or just sort of natural population growth. And, from what we see with this F4 statistic is that in all cases, the Medio period individual and the Viejo period individuals that clade doesn't break. They stay more closely related to each other than either does to any of these test populations. And that's reflected in this Z-score here. So in order for this clade to break, in order for, Medio period individuals or Viejo period individuals to be more closely related to Chaco or Anzic or Caribbean, again, we test all published populations here, you would need a Z-score that was either less than -3 or greater than +3, and we don't see that here. So, for example, if there was a big influx of people between the Viejo period and the Medio period, and those people were coming from, say, further south in Mesoamerica or further north, this clade would break. We would have a Z-score, that was either significantly less than -3 or greater than +3, and we don't see that. It's one line of evidence, but it is, maybe providing some new insight into some of these longstanding archaeological questions about Paquimé and the people who lived there. And this is just another way to visualize this. It's a different form of the F4 statistic. In this case, what I did was take one type of ancestry, so that Tiopa ancestry, northern, that's sort of the maximized northern Mexico ancestry that we have in the data set, and then that late California ancestry, just to see how all these different test populations, where they fall along that continuum. And as you might expect, the both Medio period and Viejo period individuals are down by this Tiopa ancestry. Again, if there was some influx of people between those two periods, we might see, we might see Paquimé Viejo down here, and then Paquimé Medio period shoot up here, but we don't see that. There's not some group of people moving into Paquimé that had distinct ancestry arriving at the site and mixing with the people who lived there between the Medio and the Viejo period, at least initially in our preliminary data. Okay, so as I mentioned, this is meant to give you an example of what we can do with ancient DNA. The big Pippanom paper, my goal is to have that out by the end of next year, so there's plenty more to come. But I would love to answer some questions that people have. Before I do, my acknowledgements, the authors on this paper that just came out: Meredith Snow, Mike Searcy, Jose Luis Punzo, Steve LeBlanc, Frank Ramos, Lori Eccles, and David Reich. As I mentioned, ancient DNA really is collaborative work. This is actually a small number of authors for an ancient DNA paper. Usually there's dozens or even hundreds, but it's because these papers, not only require wet lab expertise, analytical expertise, archaeological expertise, but then also, if we bring in radiocarbon dating or isotopes, all of those people need to be included as authors too. So for me, that's been really one of the pleasures of ancient DNA is meeting people who have backgrounds and specialize in fields that I don't, and learning from them. But I also wanted to thank the Consejo de Arqueológica of INA. They gave us permission to conduct this study and collect samples from these hundreds of individuals across northern, western, and central Mexico. definitely want to acknowledge the ancient people we study and their modern day descendants. All of our PIPANOM collaborators, archaeologists, and researchers across Mexico. The key funders for the project were the Howard Hughes Medical Institute, John Templeton Foundation, and National Geographic. I can't list all of the Reich Lab technicians and bioinformatics group people who generated this data for me to study. And then the Old Pueblo Archaeological Center, Al and AJ, it's really my pleasure to talk about this topic and share some of these experiences that and this knowledge that I've acquired, as a researcher in the Reich Lab in the ancient DNA world with this archaeological audience. So yes, I'm happy to answer questions and chat more about some of these preliminary results that we have.
It's a really amazing experience that you've gathered and developed, and we're really delighted to have you speak with us. It seems like these techniques have the amazing ability to answer questions that all archaeologists have about movement of people, and also give new insights into questions of power and status and all different types of avenues that everyone's looking at. So thank you so much for being with us. I think before we pepper you with all sorts of inquiries, I think that Al has a few things that he wanted to share really quickly.
Yeah, thank you, AJ.
Mhm.
Just some words about Old Pueblo Archaeology Center and what we do. Our mission involves educating people, children and adults, about archaeology and other cultures, fostering preservation of archaeological and historical sites, and developing a lifelong concern for the importance of non-renewable resources and traditional cultures. So some ways in which we implement our mission: we offer educational tours to sites of archaeological and cultural interest. These are just a few that we have coming up between now and February. We do have some more spaces open on the Badger Springs tour now. And on the San Pedro tour that's coming up in January, that one's filling up pretty fast. The other two have more spaces available here. We offer classes and workshops, a lot of online classes. I usually offer at least 3 online classes per year on Southwestern archaeology. And we also have an arrowhead making and flint knapping workshop coming up for those of you who might be close to the Tucson area on Saturday, November 9th. We do have an online presence. Our next Third Thursday Food for Thought program will be a program on history of Arizona, especially Southern Arizona, about unsung heroes, search and rescue, and first responder personnel, the history on that topic. This will be by Butch Farabee, who is a retired National Park Service, eventually a superintendent, but did a lot of search and rescue during his career, and he's quite a fascinating speaker on this. We do a lot of upcoming activities, email announcements. So those of you who've registered for this program and haven't been getting our emails previously, we will add you to our emailing list to receive announcements about upcoming activities in the Southwest. We send these out twice a month, and if you don't want to receive them, you can unsubscribe anytime you want. If you do want to receive them, you can go to our homepage here, oldpueblo.org, and enter your name and email address towards the bottom of our homepage, and you can get onto our emailing list. We do a lot of children's education programs. We have a simulated archaeological dig site set up at our facility in Tucson, which is on a property of the Tucson Unified School District. And most of the money that we take in from grants and donations and memberships goes into this education program. So we strongly encourage people to make donations, which you can do here by sending us an email or taking a look at our website. And speaking of children's education, this Saturday, we have a program coming up in Tucson at the Vista del Rio Cultural Resources Park in Tucson, which is on the site of a preserved archaeological site. So we'll be offering a lot of children's education activities there, participation activities. And we can still use some volunteers for that. So if any of you are in the Tucson area and have some time to spend Saturday between 9 and 1, we'd be happy to have you help us out on that. we are kind of passing the hat figuratively tonight, like we do on all of our Third Thursday programs, asking people to participate and to make donations. You know, we are a nonprofit organization, so we really rely on donations and memberships to support our programs. And I'll put in a list in the chat again. I put it in early on of places, you know, where you can find us online. So with that, I would like to take Q&A time. Thank you.
Okay, you ready for these big questions?
I am.
All right, okay. So I'm just going to read them in the order that they came into the Q&A session. The first one's from Mary, and it says, have you compared this DNA, I think from the PIPANOM project, with DNA from the US Southwest?
So at this point, the published data from the US Southwest is from Chaco Canyon, really, paper that was published in 2017. And then those mitochondrial data that I mentioned earlier. So I have looked at the mitochondrial DNA. That's a little bit more straightforward because really all we're doing is looking at those different haplotypes. There's a little bit more, a few more complex things that you can do. And then, yeah, with the Chaco data, you saw some of that in the slides that I had. And you know, my advisor in one of my advisors in grad school was Steve Lexon, who has written extensively about the relationship between Chaco and Paquimé. So of course I was interested in that. And you know, I sort of hinted at not really finding evidence for a migration from, you know, Chaco into Paquimé. I will say there's a huge caveat with that, which is that, you know, the individuals from that 2017 paper, the Chaco Canyon paper, were elite individuals. And, you know, if Steve's hypothesis is correct, it's only a small percentage, you know, those elites that moved into Paquimé and then maybe, you know, founded the site or had some sort of power influence there. And so we wouldn't, if there was, we wouldn't necessarily expect to see Chaco ancestry, you know, widespread throughout Paquimé. We'd actually only expect it to be in a handful of elite individuals. Now, I'm arguing that the child burial 23E is an elite individual at Paquimé, and that individual, at least the ancestry doesn't look like Chaco to me. Chaco's distinct enough that I can distinguish between Chaco and Paquimé. But there's some other techniques that we're going to try. You know, first of all, we need to make sure we're analyzing elites from Paquimé. We know who are elites and we know who are commoners, and we haven't really done that work yet in our data set and the metadata. But there's also a new technique that I didn't mention that's kind of related to ROH called IBD. It stands for Identity by Descent. And it's a new way to look at how closely related individuals were. And that's really the technique that we need to use to see, you know, relationship of any individuals from Chaco to Paquimé or any of these sort of cross site comparisons, because that allows us to see individuals that were more distant related, which is what you would expect. You know, Chaco and Paquimé are separated by, you know, 300 years, 250 years. And so looking at the ancestry components, you know, you might expect there isn't necessarily the best way to do that because of the time gap. But if we use IBD, we might be able to distinguish that. And haven't done it yet. I'm not, that's one of those techniques that I haven't learned yet. And I'm going to have to ask someone in the lab to do for me because, as I mentioned, I learned all this stuff sort of on the fly. If I would have had my druthers 12 years ago, I would have just been digging holes in the ground. So some of these more advanced techniques are a little above my pay grade, but that's the one that I really want to do to sort of explore those questions.
Sounds good. The next one's from Cynthia, and it says, how likely is it that two individuals with high ROH were deliberately bred as sacrifice, or is that actually impossible to know?
So that's getting in. I can tell you genetically that an individual had parents that were closely related, but genetics won't tell us why those two parents had a child. And we mentioned in this paper, it could have been an illicit relationship. It could have been, two people that were closely related, procreating and creating a child that shouldn't have existed. Now, I don't think that's the most likely explanation, because if this child was, sort of not so was two people, pairing together that they wanted to keep discreet, you wouldn't expect that child to be buried in this really important ceremonial building in an interesting, unique context. So that's where we have to use all these other archaeological pieces of evidence, ethnographic pieces of evidence, to help us understand some of the why. But at least genetically, I can't tell you that. That's certainly a hypothesis that I considered. It's not out of the realm of possibility. But the type of DNA data, at least that we're looking at, can't really get into that as a possibility.
And what was the age on that child?
So somewhere between 6 and 10.
Okay.
Yeah.
The next one comes from Mary. Have you used this DNA study to look at migration?
Oh yeah, that's one of the key questions that people use ancient DNA to look at. So the Americas are critically understudied compared to other parts of the world. There's a variety of reasons for that, ethical, important reasons. But I think from a scientific perspective too, there's been not enough attention given to the Americas. But if you look at papers in that are coming from Europe, some of the earliest big ancient DNA papers, so papers that date from 2015, 2016, 2017, were all about migration. And some of the the most well-known ones looked at movement of Eurasian steppe Yamnaya people into horse riders, into and across from east to west across Europe. The spread of the Bell Beaker culture, genetically through Western Europe across into Great Britain. So yeah, that's one of the key questions people use ancient DNA to look at, and one of the things that we're hoping to do with the PIPANOM project.
Very good. The next one comes from Tina, and it says, does your work with membranes and relations to northern Mexico include haplotypes? Are any other insights with membranes?
Yeah, well, that would be, that's what I'm most interested in, because it would combine both of my research interests. What I can say, based on those mitochondrial DNA haplogroups, is that, and I think I mentioned this during the talk, things are kind of a gradient. So, we talked, I just a little bit ago talked about, movement of, people from Chaco Canyon or elites from Chaco into Paquimé. But another major hypothesis is, at the end of the Mimbres Classic period, which is 1130 or so AD, people started migrating south from the Mimbres into Paquimé. And there's certainly overlap in the mitochondrial DNA haplogroups to suggest that. Now, the resolution that we have with mitochondrial DNA isn't anywhere near what we would get with genome-wide data, but we just don't have that genome-wide data for the Mimbres region. So yeah, at least according to the mitochondrial DNA, we can see that gradient, and where there's definitely overlap between the haplogroups that we're seeing in the Mimbres and what we see at Paquimé.
All right. Any more questions out there?
There was one in the chat, AJ, from John. It kind of was what Jacob was just talking about, relationships of members with Paquimé. How close is the relationship?
Yeah, I mean, I was just talking about that, and the next step would be to get genome-wide data. But like I mentioned, doing ancient DNA studies in the Americas, I think, as we all know, it's a really sensitive subject, because we have to get the DNA from human skeletal material. So, if we wanted to do those genome-wide studies, there would have to be extensive engagement and collaboration with different indigenous groups and tribal groups in the U.S. Southwest, similar to what we did in California. I'd love to do that, and hopefully one day we can do that. But at the moment, we don't have those data.
I had a quick question, and that is, if you were designing a class for undergraduates or graduate students, what do you think are the most important elements to include that students aren't getting currently to prepare them to work with ancient DNA results?
Really good question. My own personal experience as a dirt archaeologist, the things that I wish I knew. My biology was okay, but you definitely need a strong background in biology. And I honestly think probably undergrads and graduate students are getting a better background in biology than I had. But if you actually want to do the analyses, you have to know how to code. I don't know how to code. I know how to code to accomplish the things that I need to accomplish. But like I can make pretty charts, but that's just taking data and putting that data into a data visualization program, Tableau. But to actually get those statistical results, that's all on a computing environment that runs on Linux. I didn't know Linux. I know enough of it now, but coding. And again, I think probably people that are younger than me are getting that. I never had that. And statistics too. You need a pretty strong background in statistics to, understand what a z-score is, and, all these different ways that we calculate, our results.
Did you want to talk about any of the work that you're doing in terms of ethics and working with indigenous populations?
Yeah, so I talked about the research part, but that's been, more than half of my job while I was with the lab. I flashed some of the papers up on the screen at the beginning, but one of the most significant papers that came out of my postdoc in the lab was this paper that was published a couple years ago now. That was five basic ethical principles for doing ancient DNA research anywhere in the globe. And that paper really was a labor of love, because we brought together over 60 experts in archaeology and genetics and ethics, indigenous scholars, to come up. It seems like 5 principles. How hard can that be? But to get that many people with that many different perspectives and backgrounds to agree on what should be, basic fundamental things that you need to do to move forward in ancient DNA research really took. It was a lot of heavy lifting to accomplish that, but we did, and we're really proud of that, because now whether you're working in Mexico or Germany or Australia or the U.S., there's some guidelines we know that you need to follow. Prior to that, ancient DNA was, and kind of still is, the Wild West. It just kind of exploded onto the scene, and people were doing studies. Geneticists were coming into archaeology without really understanding the questions that archaeologists are asking, and the ethical things that we consider. And so, that work to bring everyone together to identify these 5 unifying principles was really important. Another really project that I was really proud of, that I was on, paper that came out last year from a site called Catoctin Furnace in Maryland. And I think I mentioned this briefly during the talk. So this was a paper where we took ancient DNA from people that were slaves or former slaves, prior to the Civil War, and after the Civil War, and worked with 23andMe and their descendants, so modern day African American communities, to link modern individuals to those ancient individuals that were slaves or formerly enslaved. And it was really powerful, to see some of these descendant people, link up with their ancestors. And the things that we heard were that because of the way that historical records have been lost and the way that African American people were mistreated and moved around, it's often hard for them to find direct links to their ancestors. And so this was a new way to do that. And again, the ethics, there's all sort of ethical questions surrounding that. And so we published, along with that paper, me and a few other people involved published a second paper that just went into the ethics of that whole project and sort of the questions that we were grappling with, and whether we should even do this study and what value it was, and all these different considerations that we had to make.
All that is fascinating. Seems like there's a couple lifetimes of work for sure for many people.
Yeah.
Any other questions for the good of the order here? All right, well, thank you so much for your time.
Yeah, this again was a pleasure. And I don't think I usually put my email address up on the slide in case people have any follow-up questions. But it's just my name, jakob.sedig@gmail.com. I'm easy to find. And yeah, if anyone has any follow-up questions, please reach out, and I'd be happy to answer them.
And they may bug you for copies of papers that some people
Sure,
don't.
yeah. Of course.
Oh, there was one question in the chat about the continuity and change California populations.
Yeah.
And I answered that, saying I can provide PDF copy of that paper if somebody wants to contact me directly. And I put my address in.
The other thing I'll say too, about the papers, if you just Google search Reich Lab Ancient DNA, that'll lead you to the lab's page, and on the lab's webpage, all of the papers that we published are there and accessible, so people can find those papers there too.
That's great. Oh, we just had a question pop up that said, what's happening with Atzlan data?
That remains to be seen. That's one of those things that we're currently working on with the Pipnom paper. So this is something with the way these types of projects are done, like the big Pipnom project, is that you'll have a paper come out in a journal like Nature or Science that will do the basic population genetics and say, like, here's what the population structure looks like. Here's how it changes over time, which to an archaeologist might not be that interesting. But one of the things that I'm hoping to do, and that we're hoping to do with the Pipnom paper, is to publish that data, do the big population genetics paper, and then have multiple follow-up papers that get into those archaeological questions. And you can kind of see that's what I'm already doing with Paquimé, right? I have an interest in Paquimé because I worked there many years ago prior to me working in the Mimbres region. But also, it's relevant to my area of interest in general, the U.S. Southwest. So yeah, I'm working on the big Pipnom paper, but while I'm working on that, there's other finer grained archaeological questions that are probably going to be published in a separate Paquimé paper. So, with the Aztlan data, hopefully the same thing. It'll be in the big Pipnom paper, and then anything that we can do to, follow up and apply it to specific archaeological questions would be great. And I hope we can do that.
All right. So with that, I guess we'll thank everybody once again for joining us for Third Thursdays. And thank you once again, Jakob, for your time. And don't forget, if you enjoyed this, to please contribute to Old Pueblo Archaeology Center. And we hope to see you next month. And have a great day.
Thank you, Jakob. Thank you, everybody, for
Thanks so much. Yep.
Have a good night.
Bye, everyone.